We're going to get started with the DeacTalks now. So the first person speaking today is Dr. Anthony Molina. He's an assistant professor in the section on gerontology and geriatrics, and he's going to be talking about bioenergetics.
Thank you for this opportunity to speak. So I'm a gerontologist. And one of the first things I learned when I entered this field almost 6 years ago is that you can have two older adults of the same age with the exact same disease burden, yet they can have dramatically different prognoses as far as disease recovery or progression. So patient heterogeneity is a central problem in all areas of medicine, but as we heard earlier, it is a challenge that's magnified among older adults. And I think we've all seen this, right? So I'm an avid cyclist. And there's times when I'm training with senior athletes out there who I can barely keep up with. But then there are others in the same age group, including those who participate in our studies, who struggle with simple activities of daily living. So this patient heterogeneity is central to really key areas of aging research. The difference between one's chronological age and biological age, and the difference between health span and lifespan. It's not just the number of years lived, but the number of years lived healthy and disease-free. So because older adults are so heterogeneous, I think there is an imperative to improve precision healthcare for these patients. Over the years, a number of studies are beginning to understand the factors that underlie patient heterogeneity. And a lot of these have been focused on individual factors like genetics, environment, nutrition, and lifestyle. But the thing to keep in mind is that all of these converge to formulate one's unique physiology. So we and others advocate that we should be searching for integrative, cumulative, this is important for aging, and measurable functional outcomes that can inform on patient heterogeneity. In my laboratory, we focus on mitochondrial bioenergetics as such a measure. So what is mitochondrial bioenergetics? Well, I'm referring generally to the processes involved in the generation of ATP. This is oxidative phosphorylation, which occurs in your mitochondria. But really all you need to know are a couple of simple things. So first is the first law of thermodynamics, which you may remember. Conservation of energy. Energy cannot be created or destroyed, but it can be shared and stored. And that's exactly what happens in cells, where energy is stored and utilized by the making and breaking of chemical bonds. So as we add a phosphate group to ADP to make ATP, which occurs in the mitochondrial matrix, energy is stored as chemical currency. And when ATP is broken down to ADP, energy is released, and this energy can be coupled to anabolism, the building of things. Now, I think we can all agree that energy is fundamental. We very literally eat and breathe for our mitochondria. 90% of the oxygen we inhale is consumed by mitochondria to make ATP, and the nutrients we just ate, like carbohydrates, provide mitochondria with the fuel they need to make ATP. Now, because energy is fundamental, mitochondria have been linked to health in all aspects of aging, from development, the onset and progression of morbidity, and also eventual mortality. And now we know that mitochondrial function is related to multiple aspects of human health, including cognitive, metabolic, cardiovascular, physical, and psychiatric. So this is what we've been doing in my lab for the past 5 years. We've been developing a blood test that can report on mitochondrial function. And the premise behind this is pretty simple, which is that blood cells are continuously exposed to circulating factors that we already know mediate systemic bioenergetic capacity. So we have now reported that by looking at mitochondrial function in specific circulating cells, we can get a readout that reports on bioenergetic capacity of the brain, the heart, and skeletal muscle. And we and other groups have shown that this blood test is related to multiple aspects of disease and age-related disorders. So this has a number of advantages. Blood tests are minimally invasive, suitable for a wide array of people and for routine use. And we're also hoping to make this affordable with new technologies we're developing. So how are we hoping to utilize blood-based bioenergetic profiling to advance personalized healthcare for older adults? Well, these are some of the current studies we have going on. We have a study focused on patient safety, whether we can use bioenergetic profiles to predict whether an individual can safely and benefit from an intervention. We're also looking at bioenergetic profile to improve outcomes. So can treatment be personalized based on a person's unique bioenergetic capacity? And then we're also looking at disease prevention, whether there are antecedent biomarkers that can be recognized before the symptoms arise. So I think that the best way to describe this is actually by using our work in Alzheimer's disease. So despite decades of research now, there are still no preventative strategies or interventions that are effective for AD dementia. AD pathology, we now know, including the development of neurological damage, occurs years before the manifestation of clinical symptoms. And therefore, the field recognizes that efforts to prevent and countermand AD rely on early detection of pre-symptomatic pathological changes. So our goal is to identify antecedent biomarkers and risk factors to predict later life AD, or conversely resilience. And to do this, we're examining the transition from normal aging to mild cognitive impairment, and from mild cognitive impairment to Alzheimer's disease. So mitochondria have been linked to Alzheimer's disease for quite a while. The mitochondrial cascade hypothesis postulated that mitochondrial dysfunction preceded the deposition of plaques and tangles that are hallmarks of this disease. And we now have multiple lines of evidence, interestingly, showing that peripheral mitochondrial dysfunction accompanies brain mitochondrial dysfunction in the pathophysiology of AD. And really, if you dig into the literature, as we've done, what we've found is that a number of AD-related factors, such as the ones shown here in the blue boxes, have been identified to affect or involve mitochondrial function, both in brain cells and blood cells. So really, our first question was simple. Can we utilize blood-based bioenergetic profiling to screen for AD risk and pathology? And these are the results of our first completed pilot study. So for this study, we recruited participants with normal cognition, shown in white and black bars, and also participants with mild cognitive impairment. We then took a blood sample, isolated peripheral blood mononuclear cells, and measured the respiration of these cells, the oxygen consumption of these cells. This is the gold standard measurement of mitochondrial function. And what we found is that 3 of the primary respirometric parameters that report on mitochondrial function, basal respiration, maximal respiration, and spare capacity, are all lower in the patients with mild cognitive impairment. But more interestingly, 2 of these 3 parameters were already decreased in patients who we identified to be at high risk for developing AD, but were still with normal cognition. So, based on this data, and really a whole lot of other preliminary data, and with support from the National Institute of Aging, we have now launched the first large-scale longitudinal 5-year study, cohort study, to utilize blood-based bioenergetic profiling. And really to wrap up, what I'm hoping that people will consider is that mitochondrial function could be a vital sign for health and aging. So mitochondria have been linked to numerous diseases and disorders, as shown in this figure. And it's my hope that one day we'll be monitoring our mitochondrial function the same way we monitor blood pressure or BMI. And I'd like to acknowledge the people in my lab who actually do all this work. I'm told there's time for 1 or 2 questions.
Question.
Sure.
Okay. Dr. Anthony, thank you for doing the research. Your intervention and disease prevention with this blood test, what's the earliest age that you could actually do the test so that we might be able to help further know when we might be in need?
I think mitochondrial function, as I said earlier, is really involved across all stages of aging. And we actually have a case study where we've done this in a pediatric study where we were able to diagnose an infant with a mitochondrial dysfunction.
Thank you for your very beautiful presentation. The mitochondrial dysfunction is present in, even type 2 diabetes. I mean, many of the chronic diseases, we have this problem. Now the question that comes is, what can you do to bring about mitochondrial biogenesis? Because mitochondria, they fuse and fission, fusion takes place, they die and they're recycled, and the new ones come up.
Yeah.
So the mitochondrial biogenesis, what effort is being made to promote that?
So I have a long answer, and I can give you that later. But the short answer is that the reason I think we should focus on mitochondrial bioenergetics is because it is something we can do something about. It's not like genetics, for example, which at the moment we can't do anything about.
Are you saying,
some of the things you could do.
Are you saying that we don't know anything about it?
No, that we can actually improve bioenergetic capacity.
Okay.
And one of the ways is, like you said, through mitochondrial biogenesis. Things like caloric restriction may be able to improve that. But we've also seen that other dietary interventions can improve bioenergetic capacity in ways that we don't fully understand the mechanism of, but we know we can improve overall mitochondrial function.
There is a compound called PQQ, okay, which is perhaps the best mitochondrial biogenesis molecule.
I'd love to hear more about it. Thank you.
Okay, thank you.